welcome you all to our monthly Genetic Code Expansion for All webinar. This is hosted by myself, John Lewick, at the University of Rochester, and Rick Cooley at Oregon State University. We're excited for today's speakers. We have Jin Kim Montclare from NYU and Ivana Nikic-Spiegel from the University of Tübingen, I think is how you pronounce that. So we'll get going because we only have an hour. So today, Jin will go first. As an introduction, she is at NYU. She's a professor in chemical molecular biomolecular engineering, chemistry, biomaterials, and radiology. She received her BS in chemistry from Fordham University and got her PhD in bioinorganic chemistry from Yale under the tutelage of Ilana Shepherds, and then did her postdoc with David Terrell at Caltech, where it seems she got bit by the GCE bug and was doing some evolving proteins with the use of non-canonical amino acids. So since being at NYU, she's focused on several translational areas of research that include fabricating protein-derived nanomaterials that include helix-elastin block polymers and coiled-coil fibers for drug and nucleotide delivery to different tissue types. She's received several awards along the way for creative approaches of her research, and was most recently inducted into the National Academy of Inventors. I don't know what that is, but it sounds good. Today, she will be telling us about a research centered around generation of nanomaterials that bear non-canonical amino acids. So thanks, Jin. Take it away.
Sorry, I had to unmute myself and then I advanced the slide by mistake. All right, so today I'll be talking about specifically our work on biomaterials and biomaterials bearing non-canonical amino acids. So our lab is interested in nature, and nature has been able to achieve these complex functions through similar building blocks, proteins. I'd like to go through some of these examples, like harnessing energy from light. These magnetotactic bacteria can sense the electromagnetic forces and then respond to that so that they can survive. And then if you look down at your hands, these are interesting biomaterials in the sense that when they're wounded, they can self-heal. And because they're comprised of these proteins, we're interested in our lab of engineering what we call artificial proteins with hopefully new emergent properties. And so if we think about the 22 amino acid alphabet, because we have seleno, cysteine, and pyrrolysine, we have these beautiful proteins that have these well-defined secondary structure that can assemble. And so, and that is how it's able to achieve its complex function. And so in our lab, what we do is we look at these proteins in nature and take our molecular scissors and cut out snippets of these protein pieces and introduce non-canonical amino acids and then paste them in ways that nature hasn't thought about. And the whole idea is if we can do this, we can achieve novel function. So the way we are able to make our artificial proteins is by using bacteria. There are living factories for the synthesis of our artificial proteins via synthetic biology. And so our lab has been interested in making these block copolymers that was mentioned earlier by John. They were made of these coil regions as well as these elastin regions. So here's an example of what we call thermoresponsive assembled proteins, but we're also introducing fluorine, and then so that would be fluorinated traps or thermoresponsive assembled proteins or traps. And so what we're doing here is we're having a sequence of an elastin, our coiled-coil domain from cartilage and oligomeric matrix protein, which I'll share with you later, and then elastin. And we're replacing all of the leucines with trifluoroleucines. And so we're globally fluorinating this protein. And the idea is we're trying to make these materials that can then be hopefully imaged. And so this was work done by a group of graduate students and postdocs shown here, Lindsay, Joe, Priya, and undergrad Cynthia, Schwan, and Erica. So like I said, we have two domains that these traps are made of. One is comprised of this cartilage oligomeric matrix protein coiled-coil. What's beautiful about this, it is a pentamer, a homopentamer that self-assembles, and it has this hydrophobic pore that can bind, in this case, vitamin D3 shown here. And there's 2 molecules of it by this crystal structure. The next domain, which is different from that alpha helix, is this beta spiral-like domain that's called elastin polypeptide. So unlike these coiled coils that form these pentamers, what the elastin polypeptide does is it assembles, first it's comprised of this glycine, valine, proline, X-proline motif that's repeated, and it forms this beta type spiral-like conformation, which then assembles and forms these twisted filaments of beta spirals. And what's nice about this domain is that it exhibits what is called a lower critical solution temperature, or LCST, meaning that it can phase separate as a function of temperature in a reversible manner. And so you can control that temperature by changing the identity of the X amino acid or the number of repeats. So what we set out to do was we set out to generate new types of materials, and what we wanted to do is we wanted to focus on cancer. So in order to target cancers, what we did was in our sequence, we engineered these RGD domains. These RGD domains can bind to integrins that are overexpressed on tumor cells. And so we've integrated those in the sequence. And then in terms of incorporating the non-natural amino acid, we're using residue-specific incorporation, where in place of a leucine, we're feeding it the isosteric trifluoroleucine shown here. And so what we do is we normally express or at least first grow up the cells, and then wash the cells of any natural amino acid, and then replenish the media with 19 amino acids plus the trifluoroleucine, and then induce. And so we're seeing protein expression post-induction in the presence of leucine and trifluoroleucine. So the first thing we did was we looked at its secondary structure, because these are domains fused together and we wanted to get a sense of what they look like. And we looked at trap and then fluorinated trap. So trap has a secondary structure as shown here in the regular lines, and then in the hashed lines, which is the F trap, we're seeing a slight loss in structure, but maintaining the conformation. When we look at the thermostability, we see a thermostability in which the F trap is a slightly more stable than the regular trap, but in general, fluorination maintains secondary structure and stability in this case. The next thing we did was we looked at its self-assembly. So interestingly, trap and F-trap forms these particles at low temperatures. And again, remember the elastin domain. The elastin domain has this LCST behavior. So as we increase the temperature, you're going to start to see aggregation happening. And that's what we see over here in both cases. And we can see that the traps have a temperature, a transition temperature that's around 40 degrees. And then the F trap, because it's fluorinated, it's a little bit more, it requires a little bit more heat for it to aggregate. So we're seeing like 45 degrees for it to aggregate. But it's doing similar things, meaning it's forming these particles, which then as a function of temperature start to aggregate. So to assess what these particles are, what kind of assemblies these particles are, we then looked at whether they formed micelles by incubating it with this 5-dodecanol aminofluorescein. And so we're seeing that in both the cases of trap and F-trap, they assemble into micelles. And we can get a critical micelle concentration where we have trap having a slightly higher critical micelle concentration, about 0.75 or 12 micrograms per milliliter. And then the F-trap showing a slightly lower CMC, but otherwise still assembling into micelles. So both of them are able to assemble into micelles, which is great. The other thing that we wanted to look at is whether these now micelles can now encapsulate the drug. So as you recall, in addition to that elastin domain, we had these coiled-coil domains. And in those coiled-coil domains, there's a hydrophobic pore. And we hypothesized that hydrophobic pore could bind small molecule other than vitamin D3. And we had done studies in the past where we looked at just the COMCC domain, and we've seen that, yes, indeed, it binds all sorts of hydrophobic small molecules and sometimes non-hydrophobic small molecules as well. So we looked at loading efficiency, and we saw that F-trap showed higher loading efficiency, about 4 times higher, or actually no, 3 times higher binding or encapsulation efficiency to the small molecule, in this case, doxorubicin. We also did a temperature-dependent release. So in all cases, free dox, F-trap dox, and trap dox have an accumulated release profile that's similar. However, when you raise it above the transition temperatures, when it starts to aggregate and misfold, or fold in a different way, we start to see burst release. So this would be another mechanism of which we can control the release of the small molecule in our micelles. But the most important part of here is that we're introducing a fluorine. And when you introduce a fluorine, we can hopefully use that as a signal to monitor via NMR, for example, or magnetic resonance imaging. So first thing we did was we did 1D NMR. We did T1 and T2 studies as well. But as a function of temperature, we see like 3 peaks and the broadening of the peaks as we raise the temperature. So when we look at T1, we're not seeing any real differences in signal. They're kind of overlapping. This is the zoom in of it. But when we look at T2, we're starting to see a temperature dependent change in signal. So this then allows us to look at fMRI, magnetic resonance imaging, using gradient echo and zero echo time pulse sequence. My colleagues at the medical school developed a coil specifically for our proteins. And so we had one layout, our phantom layout, in which one had just water in it and one that had our F-trap in it. And so when we do proton imaging, we see signal in both because they have protons. But then when we do fluorine's zero echo time pulse sequencing, we only see signal in the fluorine case. And here's our overlay. The spatial resolution is about 468 micrometers squared, and we have a signal to noise ratio that's 43. It takes about an hour, so that might be a little long. So we're now looking at exploring other fluorinated amino acids and trying to increase that signal. But this is an example of how you can use non-natural amino acids in imaging. But then what we did was we started to explore the C homopolymer. So can we make this homopolymer smarter, right? This is the actual construct that is doing the drug binding and all of the important part. So I had an excellent group of students here. I have Jasmine, Jennifer, and Rudy, who decided to see if they can make fibers out of these coiled coils by re-engineering, basing on the COMCC, re-engineering the system. And so what they did was they took a portion of the sequence following the Q residue or the glutamine residue, and then swapping it to the N-terminus, and also removing the last heptad to generate this Q. And through modeling, they found that had this nice charge distribution that would allow for potentially a self-assembling into fibrous structures. As a negative control, they made this L sequence where they took this yellow portion of the sequence, again, removed the last heptad, and shown here. And this, from their modeling, showed that it wouldn't form alpha helices. So when we take our Q protein, as they saw these fibers forming. So indeed, this Q design was able to form fibers. This is at pH 4. And the fibers ranged from hundreds to 500 to 600 nanometers, the thickness in terms of diameter. But they had these protofibers in it that measured about 3.5 nanometers in diameter. shown here. And that is the diameter of the actual independent coil, the coiled coil, the pentamer assembly. So if you look at the fibers, you see these protofibers, and these protofibers are assembling in a way that allows it to thicken, but it's comprised of these individual fibers that are made of these coiled coils. So the next thing we did, so this is just imaging the fiber in the absence of the small molecule. Again, it has that hydrophobic pore, and that's the business of encapsulation. So we thought we can encapsulate curcumin, and indeed we did. But what's interesting is we go from nanometers size fiber now to micron size fiber. So curcumin, what's nice about curcumin is its fluorescence upon binding to the actual fiber. And so we're able to see these micron sized fibers. So from sequence to structure, these forming alpha helices to forming protofibers and then nanofibers, we can get fiber thickening through mesofibers. We're having some control of length scale in terms of thickness of fibers using these design coiled coils. Now, in terms of non-natural amino acids, before I get into that, I wanted to talk about iron oxide templation. So there's the example I had shown you was that magnetotactic bacteria. What's interesting about that bacteria is that it can crystallize iron oxide nanoparticles in its interior. And those proteins that are in those magnetosomes, which is where those iron oxide particles are, is called magnetosome-associated protein, or MMS proteins. And so someone had done studies in which they take MMS and they can just, under ambient conditions, crystallize iron oxide nanoparticles, but just only the C-terminal, the short C-terminal sequence can also allow for the iron oxide to form. And so using this and being inspired by that, we decided we were going to decorate our fibers with these iron oxide, hopefully nanoparticles. And in order to do this, it's team led by Lindsay and Dustin. Here's Tiva, Kamia, Xuen, Erica, Che-Fu, and Cheng. And and there's Michael. And all of them were involved in this because it required a lot of stuff. But in any case, what we decided to do is we decided to make this CMMS6 with a propargyl glycine on the end. And then the next thing we did was we took our Q protein, but this time incorporated an azide using azidohomoalanine, and then do hopefully copper catalyzed azide alkyne click chemistry to generate the peptide decorated onto the fiber, and then precipitate using these iron precursors to make these iron oxide nanoparticle templated fibers. So this is what we wanted to do, and in order to do that, we had to incorporate the non-natural amino acid azidohomoalanine. So here's Q wild type with just methionine. Here's Q wild type in the presence of azidohomoalanine. Here's the mass spec showing that we can indeed incorporate the azidohomoalanine versus the methionine. The next thing we did was we looked whether to see they would assemble into fibers, and indeed both of them assembled into fibers. Their thickness was similar in thickness, and so the presence of having that azidohomoalanine didn't impact the fiber assembly, which was good. Next thing we did was we encapsulated doxorubicin, seeing that in the past we had shown curcumin as the small molecule. In this case, we wanted to look at doxorubicin because we're looking at cancer again. Here we have now the fibers, we're encapsulating dox, and we're able to encapsulate dox as well. And again, upon encapsulation, the fiber size thicken, right? From nanometer size fibers to micrometer size fibers as expected. Then what we did was we added BS3 to cross-link the fibers so that they would stay when we do the iron oxide templation. So we then looked at, Well, then we looked at first click chemistry. Click chemistry, where we then click the CMMM6 with propargyl glycine, and we can see post click, we see the presence of the clicked products, the shift in the bands of the fibers. And this being the excess CMMM6. So we're indeed decorating our fibers with the CMMM6 peptide. And then we looked for the templation of iron oxide. And so we added the iron precursors. We then did the reaction, and we see, that in the absence of the CMMM6 or the protein, or the protein itself, it doesn't really show iron. Well, it shows templation, but it's not it's not necessarily magnetic. And here is in the presence of same thing with Q wild type, but no CMMM6. But then, here's showing the iron oxo hydroxide intermediate. But then when we add CMMM6 to it, we're starting to see these iron oxide nanoparticles. And this is just as a control, CMMM6, the peptide itself, and we're seeing the formation of iron oxide. So when we do EDAX of them, we're confirming that these QAHA CMMM6 iron oxide particles are actually magnetite based, based on the EDAX. So we are indeed generating magnetite-based iron oxide nanoparticles. Now, with that, we can then look at magnetic resonance relaxation studies, in which we look at, as a control, ferriheme. That's the standard for looking at magnetic resonance or imaging. It's a darkening agent. So as you're increasing the concentration, what you'll see is that it gets dark, it gets black. And so the QAHA CMMM6 iron oxide should also go black as well. So when we look at the images here, we're seeing, as a function of concentration, here's 0, and then we're going clockwise, increasing concentration. In the case of ferriheme, we're seeing darkening. And in the case of our system and the iron oxide, we're seeing darkening. Not as good in terms of the T2 weighted studies. However, in the T2 star weighted studies, here's the ferriheme, which is showing darkening, but ours is showing much better darkening in terms of the magnetic resonance imaging. So the other thing we did was we injected them into mice, into the hind legs, the material on the green being the R protein bearing the iron oxide, and then the ferriheme on the right. And so, so as you can see, in terms of T2 star, we're seeing darkening occurring here, as well as in the case of the ferriheme on the right. And so, so we're able to see imaging, or we're able to image our materials. We're able to encapsulate our materials, and it's all with non-canonical amino acids. And so that's good. That's the end of my talk. Hopefully I've shared with you some insights on making biomaterials with non-canonical amino acids and how we can use them and develop and take advantage of their unique properties so that we can use them for imaging in this case. And all I have to do now is thank all the students and all the collaborators that helped us along the way, as well as the funding agency, and then your time as well. So thank you, guys.
Great. Thank you very much. That was fantastic. If there's any questions, you can put it in through the Q&A, or you can raise your hand, I think, and you can ask it. I'll start off with the first question. So here you really wanted to make some stable fibers with a transition temperature, and that you could probably just make it on the bench and then put it into tissues. But you discussed the time it takes, like, and you can have like for these structures to basically dissolve away, right, to become unstructured.
Okay.
that depends on temperature.
All right. So that's the transition temperature. So yes, the transition temperature. Right. So that was with the work with the fluorinated traps. And what happens is as you elevate the temperature, it's supposed to come together. And yes, so it's not necessarily, I guess it's not degrading the protein, but allowing it to unwind so that we can release whatever is attached to it.
So I guess my question there is that sometimes you want really slow release so you can get it to the tissue before it unravels and then deposit its cargo.
Right.
So right. What have you found dictates the time for unraveling?
Oh, so in the case of at least the trap or the F trap, it's just dependent on how much, I guess, the temperature. And then in terms of incubation time, we incubated it like for a half an hour and then let it and then have it happen. Obviously, if we're thinking about it in terms of in vivo applications, it's a little bit different in the sense that it would have to be, I guess, within a tissue that can be externally heated. If we're thinking about cancer tumors, I don't think it would work well with brain because that wouldn't fit, but maybe any type of external, like any type of cancer that's not entrapped or too far away, for example. And then you would apply external heat. In the case that we have, the transition temperature then shouldn't be too high, right? So if you subject the body to really high temperatures, that's also not a good idea. So oftentimes we think about trying to make sure that the transition temperature is within hyperthermic temperatures of the body and not too far beyond that, because that would destroy everything.
So is the time of unraveling related to the time it takes to unravel related to the time it takes for it to create a structure? And so are those two intertwined? And so that you can
right. So the structure is, if you looked at the data, the structure of the micelle itself if we're not, is pretty stable, right? Over time, up until 40 some odd degrees. And then when you apply heat, it's supposed to start to come together and unravel. And so like I said, it should be somewhere an external source of heating for that to happen. But yeah, the time, I don't know. I mean, we haven't done it within tissues, right? But that was just an in vitro experiment. So it might vary because also there's circulation. There's yes. A lot of undergoing, at least in an animal when I think about it. So yeah, those are things that we need to be concerned about and optimize for at least delivery of therapeutic.
Great. So there's some questions online. Okay. Mark Dumont is asked, does the trap protein unfold, say, based on CD at the transition temperature?
Right. So yes, that's an excellent question. Yes, the trap it doesn't fully denature or unfold completely, but it does start to unfold at the transition temperature. You see loss in structure. And if you go above 50 degrees, and then it's irreversible. So within the transition temperature, it unfolds and you can have it come back and it's pretty reversible. But as you elevate it even higher, it's not really reversible.
Great. Another question. Thanks for the great talk. This is from Anonymous. How do you select a non-standard amino acid to incorporate in your macromolecules? And what tools do you use or engineer to incorporate those amino acids?
Right. So selection is based on what our target. For both cases, the selection had to do with imaging, right? So I have a collaborator who's excellent at imaging. And so we were like, okay, what can we do to image? Fluorine is a signal that's very good for imaging. So that's why we decided to do fluorinated amino acid in case of the trap. And then iron oxide, that has to do with templation. So for us, it was just a strategy incorporating the azidohomoalanine was a strategy to conjugate that iron oxide templating moiety, the peptide for the reaction. So it's dependent on the application for us. And
butter?
Yes.
Or not right now.
Oh.
You can butter with this if you want.
He's talking to his kid. Hold on.
Oh, okay.
Go ahead.
So the application is for us has been imaging, being able to image it non-invasively in general. And so that's why. And then for that, residue-specific incorporation is our approach, because you can get multiple levels of fluorination or multiple levels of adding azidohomoalanine so that we can click on the peptide. And so that's why we use our approach, and that's why the application.
Great, thank you. And then Ryan, did you have a quick question? We have a real quick one.
My quick question, Kim, great talk. Be great to catch up sometime. But the one thing I didn't know was the field for protein therapeutics seems to be moving towards what would be considered, I guess, homogeneous materials, right? From sort of an FDA sort of standpoint. And I didn't know, obviously these are amazingly important steps forward with your sort of, we'll call it, residue-specific incorporation with new properties. Have you seen blowback from a therapeutic standpoint? Because now you inherently are always going to have a heterogeneous mixture with an undefined number of amino acids in an undefined number of positions. Like how has the therapeutic field felt about that heterogeneity?
That's a good question. So my perspective, like yes, obviously there are people in the community who think this is not the way, which is fine. It doesn't have to be the way. There are other ways. But I view it as a polymer. I view my materials as polymers, and polymers are inherently heterogeneous in general. And you have FDA approved heterogeneous polymers for that gel that do all sorts of different things. So I see it as a more homogeneous system with respect to polymers in comparison to polymers for vehicles for delivery, et cetera. When you're thinking about a therapeutic that is like an antibody or that is an enzyme, then that's a different question. Then having heterogeneity on the actual therapeutic component, which is the doing the business, then that's a problem, I think. But in terms of a material, I think it's not as different as a polymer system.
Great, thank you so much.
Welcome.